The series of electron carriers we had to make a list of. Correct? So far, what do we know? Tell me the complexes which will carry electrons and their names in electron transport chain. What is the name given for complex 1? NADH, dehydrogenase. Complex 2 is succinate dehydrogenase. Complex 3, cytochrome BC1 complex. Complex 4, cytochrome C oxidase. Alright. So if we don't enlarge or zoom into the complexes to see what other electron carriers are there inside that, we can just remember this sequence. So tell me the sequence of electron flow from NADH to oxygen. NADH. Don't tell the name of the complex. Don't tell the complex number. Tell the complex name. NADH. NADH dehydrogenase. Then ubiquinone. After that, no. Cytochrome BC1 complex. Then cytochrome C. Then cytochrome oxidase. Then oxygen. Questions have appeared multiple times, and it is more likely to appear where you have to identify the correct sequence of electron flow, correct sequence of electron carriers. Alright, so we will go a little bit inside and see what are the other electron carriers that we will find. Today I have increased the size even more, font size. If the electron donor is NADH, what is the sequence of complexes which will, what is the sequence of carriers for electron? I have tabulated here. This is the sequence. Now using this, you can do the redox reactions. So if you write NADH here, NADH gives electrons to FMN. So NADH is here. You just write all of this above, one step above. So here it will be FMN. Then here it will be FAS cluster. Then here it will be ubiquinone, cytochrome B, like that. So what happens? NADH is giving electrons to FMN. FMN is getting reduced. NADH is oxidized. Then FMN is here. FMN gives electrons to FAS cluster. Alright, wait, I'll just show you. To make sense of what you see in your textbook. Alright. Let us say we'll write it here. FMN, FAS, ubiquinone, cytochrome B. Alright. NADH is giving electrons to FMN. Then FMN gives electrons to FAS. FAS gives electrons to ubiquinone. Ubiquinone gives electrons to cytochrome B. You see this? This is what is written in the textbook, no? Not visible. Okay. Better? Alright. So, what you see in your textbook when you open the NCERT, it is given like this. All this upward arrow, downward arrow. So, whatever is on the left side are the ones which are getting oxidized by giving electrons. The ones that are given on the right side are the ones that are receiving electrons and becoming reduced. Now we know the entire story of oxidation reduction, the sequence of electron flow and all that. Representing it in this way becomes easy. NADH gives electrons to FMN, gets reduced. FMN is reduced. Then FMN gives electrons to FAS. FMN gets reduced, FAS oxidized, FAS gets reduced. Goes on like that. Till in the end where you reach a point where this one, it will be this one, CUB, copper centers here. This will give electron to oxygen. Alright. Oxygen is reduced in the end. Oxygen is the final electron acceptor. Oxygen gets reduced and everything else continues. So it's a sequence. So you can make sense of what is given in the textbook when you know this sequence of electron carriers. We know certain things already. Complex 1, NADH, we know NADH is giving electrons. We told it gives it to complex 1. But there are two things in complex 1. There is FMN inside complex 1 and FES clusters. Iron sulfur containing clusters these are. Okay. They will carry the electrons. Then ubiquinone is also referred to as coenzyme Q. Acetyl CoA. Coenzyme A. Coenzyme there is this one, ubiquinone. Coenzyme Q. This is ubiquinone is referred to as coenzyme Q. That we know is not a part of any complex. Therefore, there is nothing corresponding to that here. In complex 3, you have cytochrome BC1 is what we call it. Cytochrome BC1 complex is what we call. So, cytochrome B comes first, then comes cytochrome C1. In between, you have FES here also. You have FES here also. Cytochrome C, we already know. AA3 is a part of complex 4. You don't have to bother about this yet. Alright? Why I have given this is because you should know that iron, sulfur and copper are a part of this electron transport chain. Alright, questions have come related to which of these are present in the electron transport chain. Iron, sulfur and copper are a part of electron transport chain. To make you appreciate that, I have given you this, which is associated with complex 4. Otherwise, you will not be given these two. Copper centers you will not be asked in the sequence of electron carriers. That is not given in NCERT. No? So, generally, the factual things that you have to recall, whatever you have to recall will be limited to what is given in NCERT. That is why I tell you repeatedly, whatever examples are given, whatever terminologies are present in NCERT, whatever names, the language of the subject that is in NCERT is the one that will be limited to in terms of recall based questions. Questions of things that, let's say there are 48 students here. I am supposed to create this is a socializing class, let us assume. You all get time to talk to each other, understand each other and know about each other for 2 months. After 2 months, I will give you an exam based on your understanding of each other. I cannot ask about somebody who is not a part of this class. I can ask about the people in this class on those aspects of their life which you might not have known. Concepts can get outside of NCERT because you can always correlate. It is related to something that is already there in NCERT. But examples, most often, especially of late, as we have come closer to 2024 exams, the number of questions with examples coming outside of NCERT has gone down drastically. So don't bother to remember all the sequence of things, so many animals, so many plants, so many examples for different types of estivation, different types of whatever, ovaries. Don't bother about that. But don't miss even a single thing that is there in NCERT. That is where your perspective notes will help. Alright? So this will not be a part of the sequence of electron carriers that will be given for you to arrange. They can ask you in terms of arrange the electron carriers present in the electron transport chain in the increasing order of electronegativity. So if they ask you about it in the increasing order of electronegativity, will you go up, down from above or below up? You will have to go from below up. Oxygen will be the first one there. If it is in the increasing order of electronegativity, oxygen will be the first one because oxygen is the least electronegative in this sequence. Alright, so pay attention. It can be questions related to arranging the electron carriers in increasing order of electronegativity, in decreasing order of electronegativity, increasing order of electronegativity, decreasing order of electronegativity in terms of which are the electron carriers. You can be asked where all do you find the FES clusters. Iron and sulfur are present in 1 and 3. Copper centers are present in complex 4. That's all. From here onwards, ubiquinone to the rest is the same even when the electron donor is Ubiquinone 2, oxygen is the same even when the electron donor is FADH2. But if you see here, another special thing that you find, ion containing heme. Heme is a part of complex 2. So complex 1, complex 2, complex 3, complex 4 have some special substances. Complex 1 has iron sulfur. Complex 2 is also ion containing in the form of heme. Heme has iron at its core. Complex 3 again has iron and sulfur. Complex 4 has copper centers. So here 1, 2, 3 have iron centers. 4 has copper center. Excuse me, somebody is repeatedly calling. Mostly it will be spam call. Hello. Hello. Yes, Heli. Hello, Heli. Wrong number. I told you. Repeated calls on my spare number, alternate number is mostly spam. But they will not stop until I tell them. Alright. You notice here, complex 1, FES, ion center, complex 2, ion center, 2 centers here, FES and heme, complex 3, ion center, complex 4, copper center. So this much should give you enough understanding. So instead of FADH2, there it was NADH, FES was same there, in between NADH and FES, we had FMN, flavin mononucleotide. Alright, rest all are the same. Okay, so iron is present as a part of two things, FES complex and heme. Iron is the center in 3 complexes, 1, 2, 3. Copper center is present in 4. Okay. Now there should be nothing that you will not be able to answer related to electron transport chain. If there is something else, we will look at it when that question comes. Shall we move ahead? Alternate pathways we completed. Which one of the following statements is correct? Hard outer layer of pollen is called intine. Outrightly ruled out. Sporogenous tissue is haploid. Sporogenous tissue is haploid. Is it correct? Sporogenous tissue is diploid. Endothecium produces the microspores. Does endothecium produce the microspores? What produces microspores? Microspore mother cells, which comes from archesporium. Alright. Tapetum nourishes the developing pollen. That is correct. So we will just explore different aspects related to this. We know that all of these are related to pollen. All of these are related to anther. So in an anther, you have pollen sacs. How many pollen sacs are there generally in an anther? 4 pollen sacs. 2 each gets fused. Pollen sac is present inside the anther, in the anther lobes. There are 4 lobes in the anther. 4 pollen sacs are present in each, 1 pollen sac in each lobe. Each pollen sac will contain pollen grains. Pollen grain is what will contain the male gamete. Ultimately, all of this is for ensuring that a male gamete is produced and the male gamete is delivered to the female structures, that is delivered to the ovary. To receive the male gamete, ovary is designed receive the male gamete, the female gynoecium is designed in a particular way. Different flowers based on where they are, how they exist, what kind of environment they are in. There are adaptations. Some will have long style, expanded stigma. Some will not have that. Some will have a broad opening for plants, from birds to enter, insects to enter. So whatever is the design for ensuring male gamete enters the female gamete is in the flower, is in the female part of the flower. In the male side of the flower, the purpose of all of this, the purpose of the anther is to produce pollen grain. Pollen grain is also not the purpose. The purpose is to produce male gamete. The hero of the story is male gamete. It's like the most important thing, the most important person is always protected. In a war, the king goes to war in the last. If there is no other choice, the king gets onto the battlefield. Otherwise, the king is important for strategizing. The king is important for guidance, leadership. Alright, like in a police department. Generally in police department, you have the IPS officers being in the leadership position. They are not on ground. IPS officers don't go chasing thieves. They don't go doing the ground level investigation as such. They are required for strategy. They are required for guidance. They are the brain of the entire department there. So pollen grain is the star here. Pollen grain is the male gamete is the star. Very small cell. There is no way to directly deliver it to the female. So it has to be protected. Cover it with pollen. Because you have to traverse the environment, you have to go through harshness of the environment, the chance of dying, the pollen grain dying is high. So numbers. Increase the number of pollen grain, produce a large number of pollen grain, so that the numbers, by the sheer number, law of averages will work and somehow there is success in reproduction. Law of averages you have to learn from nature. When the success possibility is high, production need not be more. When the success possibility is low, production needs to be in high number. Again, in a war situation, let's say you have two enemies standing in front of you. You have two enemies standing in front of you. You have a gun. Will you aim at the enemy and shoot, or will you just randomly shoot everywhere? You will target the enemy and shoot. You will aim at the enemy and shoot. But if you are surrounded by enemies all around, do you bother to take aim? You just take it and go around. Wherever the bullet goes, there is an enemy, it will hit. Okay, increased numbers. If you are a door-to-door salesman, let's say if you are selling cars, do you produce a large number of cars and take it to every door and tell people, see, look at this, have a demo and then take if you want, otherwise you don't take it? You manufacture to order, you manufacture to demand, or if you know that the demand is high for a particular car, you will keep a certain number of cars in production ready to be delivered to cater to 1 or 2 weeks of demand, so that your waiting period is reduced. You don't manufacture and then try to sell. You will manufacture to demand. When you are selling a product where the sale happens by specific interaction. But if you are, let's say, if you are selling something door to door, you are selling some detergent that you have manufactured. You have created some detergent. You want more and more people to try it. You will give samples. You don't give samples of car. You will give test drive. That too after evaluating whether the person will be able to buy your car or not. You are selling 50 lakh rupees car, and will you give a test drive to everybody who comes and asks for the test drive? You will first talk to them nicely. In the nice conversation, your attempt is to find out whether they are affordable or not. If they are affordable, then you will give a test drive. But if it is a detergent that you are selling, you will go and distribute sachets of detergents to everybody on the street. There you want a large number of people to try, and when large number of people try it, there is a possibility that some of them will like it and buy it. Alright, pollen grain is like that. When a large number of pollen grains are produced, there is a possibility that some of them can go and fertilize the female gamete. So that is the basis on which these pollen grains are produced. Large number of pollen grains. The mechanism of archegonium, archesporium, all of these are primitive or primordial gamete forming tissues. Correct? Primordial gamete forming tissues. You have that archesporial cells both in the flower. It will give rise to both male gamete formation, female gamete formation. So the origin of these cells is same. Some of the archesporial cells will become megaspore mother cells. They can also become microspore mother cells. Megaspore mother cell will give rise to the formation of female gametophyte, which is the embryo sac. The microspore mother cell will go on to produce Microcytes. Microgametocytes are the pollen grains. Pollen grains. If we look at it parallelly, there is this original precursor cell, megaspore mother cell, microspore mother cell. What is the ploidy of each of them? What is the ploidy of each of them? Megaspore mother cell and microspore mother cell? Mother cell, haploid, diploid? Huh? Diploid. The megaspore mother cells are diploid. So being diploid and the gamete has to be haploid. So in between somewhere meiosis needs to happen, no? So megaspore mother cell undergoes meiosis, microspore mother cell undergoes meiosis. Meiosis by default will give rise to how many cells? It is a two-stage process. At the end of meiosis, you have 4 cells each. You have 4 megasporocytes, you have 4 microsporocytes. So far, process is the same. Next, 3 of them will degenerate. One will undergo 3 cycles of mitosis and become the embryo sac. Here, all the 4 will undergo 1 cycle of mitosis. All the 4 will undergo 1 cycle of mitosis, only carrying out karyokinesis and not cytokinesis. Meaning what? They will only bring about nuclear division and not membrane division, cell membrane division. That means, like this one also, 1 megasporocyte underwent 3 cycles of nuclear division, mitotic nuclear division, giving rise to 8 nuclei inside the cell, inside the big cell. Alright, this fellow had 8 nuclei, this fellow has 2 nuclei. So this membranes are produced later and 8 nucleus becomes 6, 7 cells. One diploid nucleus in the center here. Here of these, one will remain as such, only one will form a membrane. So now it becomes like one free cell inside another cell. So this is what is this cell? Huh? This is a gametocyte. This is a sporocyte. This fellow is the one who will eventually become male gamete. This fellow will start giving rise to pollen tube. Understanding? Let it be that way. Now, eventually this is the embryo sac. Embryo sac has become a 7-celled mass. Pollen grain has become a 2-celled mass. More or less, mostly it is a 2-celled mass. Sometimes it can be a 3-celled mass. Alright? Eventually, 3 cells will happen. Embryo sac we have discussed. Consider pollen grain to be like this. There is one nucleus, sporocyte cell nucleus, this is the gametophyte cell. Alright, there are 2 cells. Don't bother about the names. There are 2 cells, 1 cell within another cell. This is the pollen grain. Sometimes 70% of the times pollen grain is like this. In about 30% of the times this cell would have undergone another round of mitotic division, complete mitotic division having 2 cells inside. Alright. So then the pollen grain would be a how many cell mass? Then it would become a 3-cell mass. Generally pollen grain is a 2-cell mass. Sometimes in about 30% of the times pollen grain is a 3-cell mass. Clear? Now this cell is not so strong to withstand the influence of environment. There is wind, there is desiccation, there is heat. You need to provide some coverings around this pollen grain to make sure that the pollen grain becomes hard, strong, being capable of traveling distances to reach the female. Alright, so you get some coverings. You are given some coverings. Who gives the coverings? Where is this pollen grain being produced? Where is the pollen grain being produced? Inside the pollen sac. Where is the pollen sac? Inside the anther lobes. So anther is also having coverings on its wall, no? Anther has wall coverings. Pollen sac is formed by the anther only. It is not a separate pouch. Pollen sac is not a separate pouch. If this was the anther originally, the anther develops into a particular shape, giving rise to compartments which will function like pollen sac. So pollen grains are surrounded by the wall of anthers. Anther is surrounding. There are 4 layers in the anther wall which will cover the pollen grain. Anther is contributing to the development of coverings of the pollen grain. There are 2 coverings. Covering which is closer to its body. Now this 2 cell mass, if you consider the body of the pollen grain, there is one covering immediately surrounding it. There is one more covering outside of it. So which is exposed to the surroundings. Correct? Outer layer, inner layer. The outer layer, the outer covering of the pollen grain is called what? Exine. The inner covering of the pollen grain is called intine. That's all. Now the pollen grain is ready. Alright. This is the structure. Now we will see a little bit more in perspective to understand better. Clear so far? This is clear, no? All of you are comfortable with this. Female gamete production, that is the embryo sac formation, pollen grain formation. Should look at it parallelly. There are similarities that you can see, there are differences that you see. For similarities and differences, you should analyze the reasons behind it, evolutionary reasons. Why are there similarities and why are there differences? Why embryo sac is produced from only one of the 4 cells, daughter cells of meiosis, and why all the 4 become pollen grain here. You don't need to produce too many embryos, too many embryo sacs. Tell me one thing, what will the embryo sac become eventually? Alright, let us say embryo sac is present inside the ovary. We will not go specifically to the embryo sac. Ovary will become seed. Not the ovule will become seed. Sorry, ovule. Now microgametogenesis. Microgametogenesis means what? Formation of microgamete. Microgamete is the pollen grain. Microgamete is the pollen grain. You can refer to pollen grain as the microgamete. Microspore which is haploid undergoes mitosis. How many cycles of mitosis? 1 cycle of mitosis. Complete mitosis? Is it complete mitosis that it undergoes? It is only karyocytosis, not the cell division, only the nuclear division. Generative cell and vegetative cell is produced. Generative cell and vegetative cell is produced. Both the generative cell and vegetative cell is haploid. Generative cell is the one which will become male gamete. Vegetative cell is the one which will give rise to the formation of pollen tube. Pollen maturation. Both generative cell and vegetative cell is the beginning of this. Haploid, they are haploid. Generative cell undergoes mitosis to form 2 male gametes. Why 2 male gametes? 2 sperms while the vegetative cell forms the pollen tube. Then you have the mature pollen grain, all the cells are haploid. Why 2 male gametes? One male gamete will fertilize the egg cell and the other male gamete will fertilize the central cell, polar nucleus. Alright? Okay. It's like a pollen tube formation to pollen dissemination to fertilization is like a marriage story. Alright. First time when I teach students, all of you have learnt it already, so I am just going into the process. Otherwise, let's say if you have to teach it to high school students, pollen grain is like the ready to get married groom. Gets kicked out of the house. Go get married and make your own life. I cannot take care of you anymore. That is the pollen dispersal. Now all of them set out on their life's mission to get married. Pollen grain's life mission is what? Their survival depends on whether they get married or not. They either get married or die. So whether they get married or not is dependent on chance. Is it written in their fate or not? Alright, so they go knocking on doors. They are all carrying, all of them are carrying ID cards. They have been given ID cards. It's a match. They should match. It's like a biometric ID. There should be, or it's like Hawala. You know Hawala? Hawala transactions? You don't know Hawala transactions? Illegal transactions basically. You have to transfer, let's say, 10 crore rupees from India to Singapore. How will you transport? Cash you have to transport, 10 crore cash. You cannot put it into bank account and send it via bank account. Then it becomes documented transaction, you will get caught. You have to send 10 lakh rupees to 10 crore rupees of illegal money from one country to another country. There is a network of people in every country who have that money. Now I will give that 10 crore rupees to you. Alright, you will give me a half note. You will tear a 10 rupee note, you will give half of it to me. You will send the other half with somebody else to the person, him, who will give money in Singapore. Alright, so he has the other half. I am supposed to go to Singapore, meet him and give my half of the money. Alright, I give my half of the money, he has the other half of the money, he will give it to the person who brought that other half of the money in Singapore currency. So money did not go from here to there, but my money reached him. Why should I tell my money? I am not transferring any illegal money. Somebody's money who wanted to be transferred illegally got transferred. Pollen gene is like that. It is having something which has to match. So that matching with the stigma, pollen identification should happen. It will go knock the door. Can I please be allowed to marry your daughter? Stigma says, show me your ID. Sorry, ID doesn't match. Oh, this fellow has already fallen under the stigma. He cannot get up and go anywhere else. Gone. I don't have marriage in my fate in this life. Next life, probably. So when matching happens, the stigma will open up. Please welcome. Pollen grain says, no, I am not supposed to enter completely. Vegetative cell is instructed to create a path for the groom to walk. What is the path for the groom to walk to the bride? Pollen tube. So through that king's path, Rajamarga, the pollen grains, the generative cell starts walking. But as he keeps going inside, he is told that it is not one girl who is there for marriage. There are two girls for marriage. Alright? There are two girls to be married. Mother did not think about this. But mother has given me the ability to divide. I will clone myself. Okay, pollen, the generative cell clones, divides and becomes two male gametes. Now both of them look at each other. Yes, come, we'll go. Alright. Come, we'll go. They go and they reach the embryo sac. Near the egg, there are two friends of the egg who will not allow her to get married. Alright? They are two friends. They are very close friends. They have grown up together. They are born together. They are grown up together. They say, no, we will not allow you to get married. These fellows will not live. They will kick open and make way to enter into the house. So the antipodal cells are gone. When the friends who are obstructing marriage are cleared from the path, one fellow goes and gets married to the egg. The other fellow goes and gets married to the polar nucleus, central cell. But only after this, both of them realize we are no longer what we were. Identity is gone. Egg is also no longer egg. Male gamete is also no longer male gamete. When both of them come together, they become zygote. Where is egg? Where is the male gamete? Gone. That is marriage. Identity gone. Then the marriage between the male gamete and this polar nucleus. They were having their own identity. This fellow had a king's path to come and get married. But once he marries the central nucleus, polar nucleus, it suddenly becomes endosperm. Now his job is to work hard, their job is to work hard and feed this baby, growing baby embryo, alright? And they will get used up in the entire process of making the baby grow. Correct, no? Embryo is produced. What is the role of endosperm? Endosperm, see, for making the embryo, it is only two effort. One male gamete, one nucleus, one female egg, one nucleus. Two nucleus coming together, zygote is formed. But to take care of that food guzzler, three nucleus comes together, grows so much, you open a groundnut, peanut, the entire peanut is endosperm. Embryo is sitting inside, so happily it's a small baby embryo, you have to search for it under a magnifying glass. So all that effort goes in feeding this baby. So that is the story of reproduction in plants. Nothing much different for in humans also, no? You think about your parents. They were happily going around. Maybe if you see your father's younger days photos, he might be having those hippie hair, all stylish probably. Mother might be involved in so many things. Gradually start keeping the photo of year after year post marriage, and especially photos after you came into existence, the food guzzlers. Alright. After the food guzzlers came into existence, the entire life is to feed. Not only food, feed your desires, your comfort. Now, you're all here. Every 2 weeks they have to come, give you something to feed. Correct? No, they are doing that. Endosperm, nourishment. That's all. That is the story of reproduction in plants. So, see, I tell you, everything is the same. So, when you are still having the ability to become strong enough to not just be suppliers of food to the food guzzlers eventually in life, you should make yourself strong enough so that you will be able to do other things also, other than just supply food to the food guzzlers. So, this is so much derogation of babies, no? Right. Anyways, okay, understood, clear? So, initiation, formation of two cells. Now you will not forget, two cells are formed. Microgametogenesis, maturation. Structures encountered from outside to the inside of a pollen grain. You go from outside to the inside of a pollen grain, what all do you come across? First you come across the exine. You cross the exine. Exine is made up of sporopollenin. You should remember this. Sporopollenin is most important. It is a very hard substance, tough substance, makes the pollen grain tough. Intine is the inner layer lying just beneath the exine, made up of cellulose and pectin. It's more flexible than exine. You get the vegetative cell, which gives rise to the pollen tube, and the generative cell, which divides into the sperm cells or the male gametes. Key similarities and differences between pollen grain formation and embryo sac formation. Initial cell, see, I have listed it as like this. What is the parameter? What will happen in pollen grain formation, embryo sac formation? Is it a similarity or a difference? That's all. Initial cell, what is the initial cell for pollen grain formation? Microspore mother cell diploid. For embryo sac formation, megaspore mother cell diploid. Similarity. Next stage is type of cell division, meiosis followed by mitosis, meiosis followed by mitosis. Similar. Number of functional cells produced 4 microspores. All are functional. 4 megaspores, typically only one is functional. This is the difference. Then resulting gametes, 2 sperm cells, haploid. 1 egg cell, 2 polar nuclei among others, all haploid. This is the difference. So that will give rise to 2 haploid cells. 3 haploid cells basically, eventually. 2 are gametes, here 1 gamete. Supporting cells. Vegetative cell which forms the pollen tube. Synergids and antipodal cells. This is another difference. Final structure is pollen grain, which is the male gametophyte. In females it is embryo sac, which is the female gametophyte. Similar. So, okay. layers of covering around the pollen grain. Alright. Exine and intine are the two layers. Each of them have two layers. Each of them have two layers. Both the layers of the exine are more or less similar with small binary differences in little bit of functions also. Same with the intine as well. So exine outer layer is the sporopollenin layer, provides protection, made of sporopollenin, exclusively sporopollenin, predominantly exine outer layer, provides protection against environmental stresses such as UV radiation, desiccation and enzymatic degradation. Inner layer of exine is also referred to as foot layer. Sporopollenin is the predominant component. Sometimes you can have cellulose. Structural support and connects to the intine layer, helping maintain the integrity of the pollen grain. Intine is made up of pectin and cellulose. Pectin and cellulose provides flexibility and structural support to the pollen grain, facilitates germination by allowing the germination of the pollen tube. Inner intine has hemicellulose also, along with cellulose and pectin, supports the cytoplasm and organelles inside the pollen grain, involved in the pollen grain's response to external stimulus during germination. So outer exine is called sporopollenin layer, inner exine is called foot layer. Sometimes you call it as exine, exine also. So many terms, you can, different ways of representing the same thing. Questions arise in terms of the composition of the coverings of the pollen grain. Exine is predominantly made up of sporopollenin, which is a very hard substance, and the intine is predominantly made up of cellulose and pectin, small components of hemicellulose. Okay. This one we already discussed yesterday, testa, tegmen, tectum, tapetum. Alright. Testa, tegmen, seed coat, outer seed coat, inner seed coat. Tectum is outer, the hard substance on the outer layer of the pollen grain, the outermost sculpted layer of the exine in the pollen grain. Tapetum is the innermost layer of the wall of the anther or the pollen sac. Can you tell the four layers of the wall of the pollen sac from outside to inside? Epithelium. Huh? That is epidermis, okay. Epithelium or epidermis. Then middle layers, endothecium and tapetum. Tapetum. Each one contributes some things. Got it? Okay. Done? So, related to pollen grain, intine, exine, tapetum, all of this is done. When we get a question related to anther, we will elaborate a little bit more on anther, its development, how does it form, how does the anther breaks open, all of that we will see there. Alright. We have to restrict somewhere so that we build something, let it synthesize, go to something else, build it, let it synthesize. When we come back to the same concept through some other question, the exploration will be in a different dimension. By then this would have synthesized in your brain. Alright. Okay. So, I will share this by the end of the week. Only this paper. Alright. Only this paper and physiology and this thing. Just to serve as a reference for you to build other notes also. Alright. Because I am sure that if I start giving you these notes that I make, you will stop making your notes and you will be tempted to read these notes repeatedly. Your mind will start telling you why make substandard notes that I am making anyways. If I am getting good notes, let me read from this. Instead of wasting time making notes, I will utilize time well by reading the ready-made notes. Making notes will save you. This will go on to thousands of pages. Right? If I make it, it will become thousands of pages. You will not have time to read. You have books, no textbooks, thousands of pages, having more information than I make in the notes. So, don't get tempted. Stick to the process. It is you who will become successful, not me who will make you successful. So work is not on what I give you, work should be on what you make yourself. Alright. So in this industry, in the coaching industry, there is too much importance given to the teachers. Too much importance given to the trainers. Oh, I am the one who has given rank 1 consistently for 6 years. How many students did you teach? How many students among all those you taught got a medical seat? How many students did not get it? That is the statistics you should say. If 1 out of 10,000 that you teach or 1,000 that you teach has got rank 1, 800 have not got medical seat. Even if 200 out of 1,000 have got medical seat, it's a big success of the students. Alright. Do you attribute the most common outcome to the cause or the exceptional outcomes to the cause? What is the attribution? Should I claim responsibility for 10 out of 1,000 people getting top ranks or 990 out of 1,000 people not getting top ranks? What am I responsible for if I am responsible at all? Alright. So don't give too much importance to what I will be able to do to your preparation or what other teachers will be able to do to your preparation. It will feel good. We will help you to clarify the blocks and get onto the preparation path more intensely with better clarity. We will definitely be able to give you better clarity of understanding. But most important thing that will help you succeed is what you become. We do have. You should be like one that you want to be, do what they did, and then you will have it. Alright? You get a medical seat when you are a doctor. You don't become a doctor after you get a medical seat. Doctors get medical seats to pursue a course where they get a degree legally. That's all. Unless you are a doctor here, you will not get a medical seat. So work on that. Alright. Perisperm differs from endosperm in being a haploid tissue, having no reserve food, being a diploid tissue, its formation by fusion of secondary nucleus with several sperms. Perisperm, endosperm, these are the two structures that we have to remember with respect to what? Seeds. All angiosperm seeds have endosperm. True or false? All angiosperm seeds, whether monocot or dicot, have endosperm. True or false? Huh? False, huh? So tell me an angiosperm seed where inside the seed coat you find embryo direct. What angiosperms are divided into two types, monocotyledons and dicotyledons. Monocotyledonous plants have, is there anything else other than monocot and dicot in angiosperms? Can we put all plants, angiosperms, into either the category of monocots or dicots? Can we do that? Yes. Have you seen any other category of classification of angiosperms based on seed? Because we cannot say there is nothing else. But in what we have seen, have we seen any other category of categorization for putting plants into based on seed other than monocotyledonous and dicotyledonous in what you have studied? Have you seen that? No. So either all the angiosperm plants are either monocotyledonous or dicotyledonous. What do you mean by cotyledon? What do you mean by cotyledon? Okay, what is cotyledon made from? Cotyledons are made from? From the zygote. Huh? Okay. What is the purpose of cotyledons in the seeds? Huh? Provide food. So cotyledons are reserve food material. Cotyledons are reserved food materials. They are like food cakes. Food cakes available for the embryo. Embryo needs to eat food, no? It's dried food. It's like how do people going into space expedition take food? How do people who go to North Pole expedition take food? How do people who go to long duration forest treks take food? Do they take ready-made cooked food, or do they take raw materials? They take highly concentrated canned foods, concentrated foods, cakes, like let's say something that you put in water, it will increase in volume and increase in volume, so that you eat it, you will get your nourishment. So if you want to carry food for a longer duration, where you don't get food and you have to ensure survival, you have to devise ways of compacting the food and preserving it for longer duration. That means in any given space, you should have more food to be compacted compared to what can be done with ready food. So when this compacted food is made available for utilization, the quantity of food that you will have is much bigger than what you carried. Are you understanding? You are understanding? You are carrying in a compacted form. You cannot eat that compacted form. When you transform that into a form that you can eat, it will be much bigger in quantity than what you carried. So that when you eat it, you get your nourishment. If you have to carry, let's say, if you have to eat 400 grams of food per meal, you have to stay out for 10 days and you have to get 3 meals a day, 1.2 kgs per day, 10 days, 12 kilograms. 12 kilograms. What if 300 grams of food is dehydrated and made into 50 grams? If I make it into 50 grams, it is 150 grams of food in a dehydrated form that you have to carry. So for 10 days, it is 1.5 kgs instead of 12 kgs. So your weight of carrying is reduced. When you want to eat it, can you eat it in the dehydrated form? All that you have to do is take that tablet, 50 grams tablet of your food, put it in a bowl, add water. How do you get food in flights? Like that only, no? All those noodles, upma, pastas, it is all dehydrated. They will add boiling water to the mark that they have given. Leave it for 3 minutes, food is ready to eat. Cotyledons are like that. Cotyledons are food cakes. It is like dehydrated food cakes. When you add water to it, it will become much bigger in terms of food availability, available food, which will sustain the plant for significant duration. See, it is one peanut that is there, no, for the embryo inside that peanut. One peanut is how much? We will eat at a time some 100, 200 peanuts as snacks, not food. Isn't it? We eat about 200 peanuts for snacks. That is how I and my father eat peanuts. We both love peanuts. So the only way to control the quantity of peanut we eat is by regulating how much peanut is available at home. That's all. You get a 200 grams roasted peanut, 200 grams will be completed in the shortest possible time. Alright. You get 500 grams, 500 grams also will be completed. You get 2 kgs, 2 kgs also will be completed, and both of us will be holding our heads in great struggle, and we will be struggling, but we cannot stop eating peanuts. But for that one peanut, that peanut is developed by the plant with an embryo inside and all the cotyledons there as dehydrated food. Now that one seed goes into the soil, it comes out. Till it comes out, you see the biomass of the plant that is coming out from inside to outside, whatever has grown. You see the biomass of that and see the biomass of the peanut. Which will be more? Will the biomass of the plant that has grown from that peanut till the time it has gone inside to above soil. So whatever is inside the soil, the biomass of that is more than the biomass of the peanut, no? But for all that growth, this peanut has contributed food. How? Dehydrated food. Cotyledons are that. Cotyledons are like your space food, dehydrated food. 50 grams food which can become 500 grams when water is added. Understand? And when this enzymatic activity is initiated, it is not that only the peanut is having nutrients. When enzymatic activity is initiated, organic matter around the growing plant also will be acted upon by the enzymes, and that also will be utilized. Soil also will contribute. So what are cotyledons in a plant? In a seed? What are cotyledons? Cotyledons is reserve food material. All angiosperm seeds have it. All angiosperm seeds have it. Some of them have single cotyledon, some of them have two cotyledons. Whether it is single cotyledon or two cotyledons, where are they coming from? Because it is a seed, inside which we have the cotyledons, the seed is a transformation of the ovule. That means cotyledons also should be coming from something which was a part of the ovule only, no? So what is giving rise to cotyledons? What did we have inside the ovule? On the innermost part, we had the embryo sac. Around the embryo sac, we had the excuse me. Around the embryo sac, we had the nucellus. Now, around the nucellus, we had the integuments. Then you had the funicle, placenta, and all that. Integuments became what? What did the integuments become? Integuments have what ploidy? Cells of the cotyledons will be triploid. Cells of the embryo will be diploid, not haploid. Perisperm is persistent nucellus. When nucellus persists, it is referred to as perisperm. Not all seeds will have perisperm. Perisperm is coming from persisting nucellus. Alright, so now the entire ovule we know what becomes what. Clear? That's all. So what is the answer here? Being a haploid tissue, perisperm is not haploid, perisperm is diploid. Endosperm is, we have perisperm. See, when you look at the question, you have to identify what are the terms that we have. Perisperm. What is perisperm? All of this story to make you understand what is perisperm. What is perisperm? Perisperm is persisting nucellus. What is endosperm? Endosperm is the reserve food material for the embryo to utilize during germination. Perisperm is coming from nucellus. Endosperm is coming from the fertilized central cell. The fertilized central cell becomes the endosperm. Haploid, none of these are haploid. This is diploid, endosperm is triploid. No reserve food. Perisperm, endosperm has reserve food. Perisperm differs from endosperm in having no reserve food is partially correct. Is partially correct because though perisperm will be utilized for energy as long as it is available, it is not a reserve food material. So we cannot say option 2 is completely wrong. Partially correct. But option 3 is absolutely right. Perisperm differs from endosperm in being a diploid tissue, while endosperm is a triploid tissue. Its formation by fusion of secondary nucleus with several sperms. No. First of all, perisperm is not formed by fusion of central nucleus with sperm, and perisperm and central nucleus does not fuse with multiple sperms. Perisperm central nucleus will fuse with one sperm and becomes endosperm. Alright? That's all. So we will see what all notes has been built around this. Perisperm is a diploid tissue derived from the nucellus of the ovule, whereas the endosperm is typically diploid and results from the fusion of secondary nucleus with one sperm cell during double fertilization. Perisperm often, see double fertilization, and there is one more term which we refer to the fusion of central nucleus with male gamete. Triple fusion. Why triple fusion? It is the fusion of 3 nuclei. Perisperm often persists in seeds of some plants and surrounds or is mixed with the endosperm, but it does not play a significant role in nutrient storage as the endosperm does. Perisperm and endosperm comparison. Perisperm location surrounds or is adjacent to the endosperm in some seeds, found within the seed surrounding the embryo. Origin derived from the nucellus or the maternal tissue, formed by the fusion of the secondary nucleus in the sperm cell during double fertilization. Ploidy of cells diploid, endosperm triploid. See, when we talk about it as diploid, triploid, we are making an assumption that the parent plant is diploid. Diploid. If questions arise, if question comes where the ploidy of the parent plant is not diploid, then be very conscious of arriving at what the ploidy of endosperm is. You have to look at it as 1.5 ploid. 1.5 ploidy. If the ploidy is tetraploid, endosperm will be 1.5 times that, hexaploid, 6 times. Alright. Diploid to triploid, that is 2, this is 3. So tetraploid, if the plant is tetraploid, then it will be 5. No, it will be 6, 1.5 times. Function generally does not serve as the primary food storage tissue, may provide additional nutrients. That is why the second option was partially correct. Serves as the main storage tissue for the developing embryo, provides nutrients. Significance, less common, found in seeds of specific plants like black pepper. Widespread in angiosperms, crucial for seed development and germination. Presence in seed may or may not be present depending on the species. Commonly present in most angiosperm seeds. Nutrient content typically lacks significant nutrient reserves rich in starch, proteins and other nutrients. Alright. Cotyledons develop from zygote. You are right. Zygote to embryo development. Cotyledons develop from zygote only. Alright. Endosperm provides nourishment during provides the reserve food material. Cotyledons are along with the embryo. Sorry, I just digressed from the concept. Alright. We will discuss the development of cotyledons. We will discuss the development of cotyledons along with the development of embryo. Embryonic development we will discuss. Alright. Correct it. Cotyledons development is from the zygote. The seed that we have, what we have in the peanut, what we eat, that is endosperm. Alright, that is reserve food material. Cotyledons are with the embryo, those leaf-like structures. Those are the cotyledons. Alright. Okay. Perisperm is a diploid tissue derived from the nucellus and is not a significant nutrient source, whereas endosperm is triploid, formed by double fertilization and serves as the primary nutrient source for the developing embryo in most seeds. Perisperm is less common and found in specific plant species, whereas endosperm is common and critical component in angiosperms. We completed discussing. How many questions? 9 questions. 9 questions, so many chapters. What we are left out in plant biology is plant anatomy and morphology. Most of the other chapters we have touched upon in some way or the other. Plant anatomy and morphology, anatomy is still easier. Morphology is what will trouble. Anatomy is concept based. Morphology also has concepts, but what will create problems to you during exams is examples. There are 4 chapters which will create trouble in terms of examples, which will make you feel less prepared. But if you look at the number of questions that come from these 4 chapters, you need not freak out and you can limit your preparation on examples to NCERT. What are the 4 chapters? Biological classification, plant kingdom, animal kingdom and plant morphology. These are the 4 chapters where there are hundreds of examples. Animal kingdom, plant kingdom. So how many questions are coming? The number of questions are becoming less. Number of questions specifically based. Earlier there used to be questions like they would give some 20 different names and say how many of these have a particular character. Options would be 6, 7, 8, 10. I would say get lost. I don't want to answer this question. How on earth you can remember that? It's not possible. Those are questions which serve no purpose. For that, it's okay. Let's say if you give 4 or 6 characters, if you give 6 or 7 characters and say which of the how many of the characters are common between phylum A, B, C, D, that makes sense because you are asking question based on the understanding of the concept. Each phylum, you forget about phylum, every genus will have hundreds of species. Everything will have some scientific name, some common name, some local names. How will you be able to remember examples for all of that? Don't spend time in trying to remember examples outside of NCERT. Alright? So to be covered for all the examples, in the perspective based notes I have told you, example related one small booklet you make, you will just go through for every topic, subtopic, whatever is there, you are just making note of the examples appropriately. And whenever you feel bored of doing everything else, just keep revising these examples. That should take care. If there are 1 or 2 questions that you still are not able to answer based on examples, good riddance. You are able to answer the other questions because you did not spend too much time on trying to prepare for that 1 or 2 questions. If you want to feel confident of answering those 2 or 3 questions which might be out of NCERT in terms of examples, you will lose time which will make you underperform on the important questions. It's like in cricket. You have a problem with short ball, bouncers. Alright. In a match, what do you have to do? You have 50 overs, you have 300 balls. The bowler cannot keep bowling short ball every ball. 1 per over is allowed. The second one will be a no ball and you will get a free hit. So, you are not comfortable with the short ball, forget it, don't play it. Duck it. That's all over, no? Strengthen all your good shots. If you spend more time strengthening your good shots, you will be able to perform better. Parallelly, when you have time, when you have extra time, you start working on improving your ability to play the short ball also. Example based question preparation is like that. Focus on what can give you more assured return in terms of getting correct answers. If you are confident on that, as you feel confident on that and you want just a slight change of mind, then go into this. Alright, last to do is examples outside of NCERT. That would be the last. And whatever has come in the papers, in the previous papers, those you remember. Whatever has already come in the previous papers, you remember that. Some of them will be outside of NCERT also. Very likely repetition will happen from that in terms of examples. Alright. So we'll do few human physiology questions. What do we have first? Muzzle. The H zone in the skeletal muzzle fiber is due to 3 days of complete plant, and we will come to humans a little bit. Absence of myofibrils in the central portion of the A band. The central gap between myosin filaments and in the A band. Central gap between actin filaments extending through myosin filaments in the A band. Extension of myosin filaments in the central portion of the A band. What is the answer? Forget about the answer. There are so many things here. What is this question related to? Question is related to skeletal muzzle. There are some things here. Head zone, muzzle fiber, myofibril, A band, myosin filament. See, fibril, filament. A band we have already marked. Actin filament. A band we have seen. Alright. So there are some specific reference to areas. H zone, A band are areas. There are some references to structures, something long structure, fiber, fibril, filament, all of that sound like something, it is something long structure, like a wire. So you have fiber, fibril, filament. There are two filaments that we see, myosin filament, actin filament. There is one fibril we are seeing here, myofibril, muzzle fiber. What is a muzzle fiber? Myofibril filament. We understand the structure, then we will know what head zone is. No? Forgotten? Do you have multiple pens? More than one pen? Okay. Take out all the pens that you have and keep it on the table. Oh, you will have a pouch. Okay. See, what do we have here? Which muzzle is this? There is a cross section of some muzzle being taken. Which muzzle is this, of which a cross section is being taken? Triceps. It is a cross section of the triceps that is being given here. This is the muzzle of your importance. All of you are obsessed with this probably. Biceps. Gym workout means pump up the biceps. Nothing else. In our younger days, those were the times when Hrithik Roshan came into the cinemas. He made his first movie was which one? No, not Kabhi Khushi Kabhi Gum, later. Kaho Na Pyar Hai. No, even before that. Whatever, anyways. He came in and suddenly there was a craze about Hrithik Roshan, his chiseled body and all that. But initial days, you look at his initial movies, Hrithik Roshan is like hypertrophied upper body and paralyzed lower body. Alright? You watch his, you take out, you search, not now, you will not have access to internet anyways. When you go back home next year. Search for Hrithik Roshan's first movie and watch some dance or anything where his upper body is. He used to just open his shirt and show off his upper body. But suddenly people started noticing and pointing out that look at his lower body. Chicken legs. Alright. Then probably he got it and he started working out and he created a balance in his body. Alright. Okay. Anyways, triceps is taken. So this whole thing is the muzzle. This whole thing, the boundary is the muzzle. You see so many bundles here. Bundles. What are those bundles? Bundles of muzzle cells. Bundles of muzzle cells. What do we call muzzle cells? Myocyte. Myocyte, or you can also call it as a muzzle fiber. Muzzle fiber or a myocyte is the reference to muzzle only, muzzle cell. Inside the muzzle cell, you see this, you have myofibril. Myofibril. It's like a smaller fiber which has a lot of proteins arranged in a particular sequence. Myofibril. Inside the myofibril, you have myofilaments. Myofilaments, actin filament, myosin filament. Alright. So if you are designing a muzzle, see generally what do we have in a normal cell? In a normal cell, we have all the organelles. We have the organelles, nucleus is there, and endoplasmic reticulum, mitochondria, all of these are there. Muscles are specialized cells. Muscles are specialized cells. Throughout the inside of the muscle, you have contractile proteins. Inside of the muscle cell, you have contractile proteins. You cannot just let the contractile proteins be randomly present inside the cell. You are arranging these proteins in the form of small tubes. Consider it to be tubes. Inside those tubes, you are arranging the muscle filaments, actin, myosin filaments in a particular format. So that one length, let's say it's a 10 centimeter long muscle cell. It's a 10 centimeter long muscle cell. Inside this muscle cell, you have, let's assume, 10 small tubes, fibrils, muscle fibril or myofibril. And in each of these tubes, you have arrangement of the filaments in a sequential manner. You have two fibrous bands that you find, what we call as Z bands or Z bands. They are fibrous tissue to which on either side you have the attachment of actin filament, and between the actin filaments you have myosin filament attached. Repeat this, go on continuously doing this. You have the entire muscle fibril. Muscle fibril. Not understanding, no? No. Oh, sorry. Let us say this is the muscle cell. Muscle cell, skeletal muscle cell. In this cell, every organelle is pushed outside. You see, when you see the skeletal muscles, nucleus is peripheral. Have you noticed it? Nucleus is peripheral. It is not nucleus in the center. Muscle cell will not have nucleus in the center. So inside this, I will arrange tubes. In human physiology, muscle physiology is a complex topic. A lot of technical understanding you need to have. We will have it today. This is not working. This is one cell. This is one muscle cell. What do we call it? Myocyte or muscle fiber. Muscle fiber is a reference to one cell. Fibril, muscle fibril or myofibril is a reference to cylinders of structures which will have myofilaments inside the muscle. Alright, stacks of structures. Now let us say this is one stack I am having. One stack of filament I am having. Similarly, I will have multiple. The entire muscle cell is filled with these filaments. Filaments, not filaments, fibrils. Fiber, fibril, filament. These three in an order you should remember. Fiber is the cell, fibril is the cylinder containing proteins, filament is the proteins inside the fibril. Okay. So you have multiple cylinders. Or I'll give you another example. There is a wire here, correct? Well, let's say this is the wire. This is a wire. Consider this wire. Let us say this is a phase and neutral. There is only phase and neutral in this. So how many wires will be there inside this? 2. This is one wire. This wire is a phase and neutral carrying wire. So you will have 2 separate insulated wires inside this. 2 separate insulated wires inside this. Inside each of the insulated wire, what will you have? You have copper. What do we call that? Copper wire. In individual copper wires you will have. Copper wires are bundled into phase wire or neutral wire inside this, and we have this cable. This is the cable. Let us call this cable. Inside the cable you have wires. Inside that you have copper filaments. Can I say that? copper filaments. Now this cable is the muzzle cell. Cable is the muzzle cell. What you have inside the wires are bundles of copper filaments. So those wires are the fibril. If this cable is the fiber, in the wire inside this is the fibril, the copper inside that is the filament. Copper inside that is the filament. We will look at the same diagram here. Wire or the cable, wires, copper. No, this is not this. This is the entire muzzle. I'll show you another diagram. Wait. See this? You see this? This whole bundle is what? The cable. The whole bundle is the cable or myocyte, muzzle cell or the muzzle fiber. Inside this, what do we have? Individual wires. Individual wires. So those individual wires are myofibril. Myofibril. Inside the fibril, what do we have? Copper filaments. Inside the fibril, what do we have? Copper filament. And these copper filaments are filaments, muzzle filaments. You see this? Cable, wires, filament. This is one muzzle cell. Alright? You understand? Getting it, everybody? Cable, wire, filament. Cable is the cell, wire is the cylinders that you find inside the cell. Filaments are what actually contributes the contractile filaments, proteins. Okay, we'll come back. Did you understand? Hello. Oh. Now you see this, we will go from inside to outside. The innermost will be what? See here, look at this label. Visible? Now, you see this? What did I tell you? Muzzle fiber is muzzle cell. Muzzle fiber is muzzle cell. This whole bundle is the cell. What is this red dot that you see? What are these dots you see in the cell? Huh? Not the ones inside. No. Skeletal muscles. What is the nucleus status of skeletal muscles? Peripheral nucleus is one aspect. There is something else about the nucleus that you need to know. Skeletal muscles are multinucleate. Yes or no? Skeletal muscles have multiple nuclei. They are multinucleate cells. So this is like representing the multiple nuclei. Alright. Now you see this. This is the muzzle cell or the muzzle fiber or the myocyte. 3 words. Cyte is no confusion. Cyte means cell. Myocyte is a reference to muzzle cell. Filament, fibril, fiber. There comes the confusion. Filament is the smallest. Fibril is smaller than fiber. It is like artery, arteriole. Artery is bigger than arteriole. Alright, capillary. You can consider it in those 3 lines. Fiber, fibril, filament. Fiber, clear? Muzzle cell, myocyte or muzzle fiber or muzzle cell has cylinders of filaments. Each cylinder is a filament. Each cylinder is a fibril, and inside that we have proteins which are referred to as filaments. Clear? What is this muzzle cell covered with? They are covered with cytoplasm. They are covered with the cell membrane of the muzzle. What do you call the cell membrane of the muzzle? Sarcolemma. Sarcolemma is the reference to the cell membrane of the muzzle. Muzzle cytoplasm is referred to as sarcoplasm. Muzzle's endoplasmic reticulum is referred to as sarcoplasmic reticulum. Inside the muzzle, in the skeletal muscles, there are special structures, modified structures from the endoplasmic reticulum, which are utilized for storing calcium. Calcium is essential for muscle functioning. Without calcium, muscles will fix in contraction. Alright, so calcium is necessary. Calcium is stored in what? Calcium is stored in the sarcolemma. Sarcoplasmic reticulum, in the endoplasmic reticulum. There are some specialized structures called T tubules and cisternae which will hold on to this calcium. We'll see later. So the cytoplasm and the organelles of skeletal muscles are arranged in a specific manner, specialized manner. So the names are different. You have sarcolemma, sarcoplasm, sarcoplasmic reticulum. Sarco is a reference to muscle. Alright. Now multiple cells together form a bundle. Multiple cells together form a bundle, and that bundle is referred to as fascicle. Alright. Fascicle. This is the fascicle. Around each fiber, around each bundle, around the fascicle, you have perimysium covering, specialized covering around the perimysium. Around each muscle fiber, you have the covering called endomysium. Endomysium is the innermost covering around the muscle cells, connective tissue covering. These are connective tissue coverings. Why do you need to provide so much of connective tissue support? Connective tissue is for what? Connective tissue is for supporting the tissues and cells. Why do we need to provide so much of layer by layer covering around these muscles? Starting from the cell, the muscle cell or the muscle fiber is surrounded with endomysium. Bundles of muscle cells is a fascicle, and that bundle is covered with perimysium. The entire muscle is covered with epimysium. Epi, peri, endo. Epi means outside. Epi means around. Peri is also around only. Epi is the outermost covering. Peri is around a bundle, around the bundles. Endo is around the fiber. So three connective tissue coverings will protect the muscle. Protecting the supporting and protecting the muscle fiber is the endomysium. Covering and protecting the bundles of muscle fiber or the fascicles is the perimysium, and protection covering the entire muscle is the epimysium. Epimysium surrounding the entire muscle. Alright, three coverings you should know: epimysium, perimysium, endomysium. Three levels of arrangement you should know. So if we go from muscle, from outside to inside we are going. Let us see if you have understood. Excuse me. Let us assume we are entering the muscle. We have to reach the inside of one muscle cell. We have to reach inside of one muscle cell and finally go and touch the myosin filament. What all do we see? What all do we encounter is what you have to tell me. Now there is muscle. What I am first encountering? Epimysium. I cross epimysium. Where do I enter? I cross epimysium. I enter perimysium, which is covering the fascicles. I cross perimysium. What do I feel? What do I see? Endomysium, which is covering the muscle cell. I cross endomysium. What do I see? I encounter the muscle fibril. Muscle fibril. I cross the muscle fibril. Now I am in the place where I see actin and myosin filaments. Is this clear? Now with this background, we will look at all the content. Muscle fibers. How do muscle fibers become muscle? Alright. Muscle fibers, also known as myocytes or muscle cells, are elongated cells that form the basic unit of muscle tissue. These fiber groups surround together. These fibers group together into bundles called fascicles. Each fascicle is surrounded by a layer of connective tissue called perimysium. Multiple fascicles are bundled together to form the entire muzzle, which is enclosed by another layer of connective tissue called the epimysium. Hierarchical structure is for the purpose of ensuring there is maximum efficiency in contractility. There is better contractility provided. This we saw. What are these? These two? Blood vessels. Why the color difference? Artery and vein. Alright. From outside to inside we saw epimysium, perimysium, endomysium, muzzle fiber or the myocyte, then myofibril, sarcomere. Sarcomere is the smallest functional unit. Contraction relaxation happens at the level of sarcomere. Alright, we will see how that happens. Myocyte or muzzle cell. Are you able to make sense of these diagrams now? This is the entire muzzle cell. Why is it like that? See, you see this nucleus, peripheral nucleus just being pushed towards the outside of the cell membrane. You see the endoplasmic reticulum. How is it surrounding? Mitochondria. Large number of mitochondria are embedded around the myofibrils. Alright, within the muzzle. See, when you are filling up the entire cell with cylinders, every other organelle has to position itself around these cylinders only. And because muscles require good amount of energy during contraction and relaxation, ATP should be embedded around these fibrils. Alright. See the endoplasmic reticulum here. Myofibrils. These are the myofibrils. Looks like a pack of cigarettes. Correct, no? Now if we correlate it to a pack, not pack of cigarettes, we will correlate it to a pack of BDs. More like muzzle. Alright. Okay, if we correlate it to a pack of BDs, what is the pack? What is the entire pack? Oh, we get the best example. Wire, forget wire. We will use BD. From now on, I'll use BD. Alright. If we look at muzzle in correlation with BD industry, alright. What is that BD? Each BD is fiber. Huh? Each BD is fibril. So the tobacco inside the BD is filaments, actin and myosin filament. That is the active part, no? That is the active part. What is important there? Alright. So the one pack of BD is muzzle fiber or the muzzle cell, myocyte or the muzzle cell. So a box of 10 BDs is fascicle. Then a pack of 10 BD packets or a box of 10 BDs is fascicle. Then a box of 10 packets, 10 boxes is muzzle. A carton box is the muzzle. Alright. So what is the carton box packaging in the muzzle? The carton box packaging in the muzzle is what? These sessions are getting recorded. Alright. So we have plans of putting up these lectures online for the benefit or the misfortune of students otherwise also. Alright. Okay. It's a wide angle lens, I cannot escape. Alright. So carton box is what? If carton box is the muzzle, if the entire carton box is the muzzle, I could have taken the example of chalk piece boxes also. I committed to BD boxes. So now I cannot go back because it is on record. So we will continue with BDs. Don't smoke. This is just for example and the representation purpose. Alright? Okay. The carton box is muzzle. So the carton box material, the carton is what? Carton is? Correct, tell me, lovely. Epimysium. Carton is the epimysium. We open. We have a box of BD packets. That is fascicle, and the covering of that pack of 10 is perimysium. Open it, we get a BD packet. The paper covering of that is endomysium. Open it, we go inside. Each BD is myofibril. Open it, we have actin and myosin filament. If you make it multi-compartment BDs, then each compartment would be sarcomere, functional unit. Alright? Functional unit of BD is tobacco. Likewise, functional unit of the myofibril is sarcomeres, filaments. Aha, great. See, this is filament, not filament. This is the fibril. This is the myofibril, each BD. This is the whole packet. And we will go back now. This diagram. See, this is the carton box. This is the pack of 10. This is the BD packet. And inside we have individual BDs. Muzzle. That's all. Okay, structure is clear. Epi, peri, endo, muzzle fiber, fibril, and filament. See, this one is important here. T-tubule, sister name, triad. T-tubule is important. T-tubule is the part of the endoplasmic reticulum of the muzzle, or T-tubule is a part of the sarcoplasmic reticulum which stores calcium. Have you ever experienced that during night you wake up with a catch in your calf muscles? Very terribly painful. In a few minutes it will automatically settle down. So that happens because of calcium deficiency. You go to a doctor, the doctor will prescribe you calcium tablets. Alright. So I want to tell you all something. You can inform your parents. We can do it from our side, but it's better to be done from parents' end. 3 supplementation should happen for you all. Diet I have evaluated, decent. You are getting adequate proteins, like you are getting meat, egg, lot of dal. Protein adequacy is there. Calorie sufficiency is there. The frequency of meals that you are getting is also adequate. It is all fine. But 3 nutrients, three things that needs to be added to the diet, which cannot be managed in a food preparation format. It will become very difficult to manage it in a food preparation format, which you have to add in your diet, add in your routine, and ask your parents to get it. Number 1, you should ask for multivitamin, multimineral tablets. Alright? Each tablet will cost 1.5 to 2 rupees, generic tablets. That's not expensive, but it is very difficult to ensure adequate supply of vitamins and minerals through food. For that you will have to have about 5 servings of fruits every day, 5 servings of fruits. I asked for places where I get fruit here in Nongster. Apparently fruits are not available. There is not a habit of eating fruits regularly. But fruits are what will give us vitamins and minerals. We cannot escape that. So take 1 vitamin mineral tablet alternate days. That will supplement whatever deficiencies are there in the diet. 1 multivitamin, multimineral tablet on alternate days. You can tell your parents to get it from Janaushadhi, or any medical store will have it. It is an over-the-counter drug. Multivitamin tablet. 1 every alternate day. Second one you have to add is some fiber. You will get husk tablets. You will get in tablet form some husk which will help you keep the gut clean. One tablet of any fiber. You can get those Kayam Churna tablets or Pet Safa tablets, whatever you will get. You will get just simple psyllium husk tablets in medical stores. One fiber tablet in the night every day. Important. And I also noticed that the diet does not have enough probiotics. Probiotics are very important. Probiotics are fermented foods. There is no habit of having fermented foods so much. Curd or idli, dosa, all of those are fermented foods, not a part of the natural daily food habit. So one tablet of probiotics, it can be a tablet of sporolac, lactobacillus, one tablet in the night. These three should be added. Otherwise, over a period of time, there is a possibility of you falling ill. Add these three to your diet, your immunity will get boosted, your alertness will be better. Gut health is directly related to clarity of thought. If your gut health is not right, your thinking will be clouded. You will be drowsy, you will feel sleepy, you will not be able to sleep well, you will not wake up refreshed. So one tablet of probiotic, any probiotic, so sporolac or something, can do. We can do it as a whole. We can get tablets for everybody and do that. It is very difficult to manage that. So it is better that when your parents come, they get it for you. You manage your own routine. Or you can get written consent from your parents. Then we will arrange for it from the medical college or somewhere. Alright. But these three needs to be added to the diet. These three needs to be added to your routine. I told you because of what? T tubules, calcium. Calcium is where we deviated from, and calcium is very, very important there. Why calcium is important? Where in the muscle physiology calcium comes into play? We will see that when a question comes about muscle physiology. This is a question about muscle anatomy. So we will stick to that. If we go into that functioning also, it becomes very complex now, and you will get mixed up. Right now, remember T tubule. Alright. This also is, now we will come to sarcomere. Now you see this is the Z line that we talk about. You have to attach it, no? Consider you need to put pipes in this room. Pipes in this room, and it should continue for about 100 meters. You have to put pipes. Can the pipes float in the air? Can the pipes float in the air? No, you have to have something to hold the pipes. Let us say this is the muzzle fibril. This is the cylinder, muzzle fibril, in which we have to arrange the muzzle filaments. We have to arrange the actin and myosin filaments. And they are all like rods that we will have to put here. Rods which can be used to control the movement of these walls. Alright, this wall, let us say. And this is a continuity. The cylinder is continuous. The myofibril is continuous. I am talking about one unit. So let's say we have a connective tissue put here from one end to the other end of the filament. Not the filament, the fibril. Another end also we put it. So we put some small rods from this side to that side. So if this is the wall, visualize it. This is the wall. This is the wall. I am putting some rods like this. Actin filaments I am fixing like this. Now will the actin filaments stay in place? Will the actin filaments stay in position? They do stay in position because I have put a wall. I have put a connective tissue wall to hold it. You see this? Z line. This is the Z line. This is a connective tissue. This is the connective tissue that is holding the actin filaments in place. These are actin filaments. Which ones? The pink ones, thin filaments. The pink ones are actin filaments. Alright. Clear? Now, I am putting another wall on the other side also. I will put one more wall here. Same I will do. We need to ask for more pens. Are pens not there? All the pens that are there in the drawer is here. I'll write it in black only. Oh. Madam, it's not there. Madam, red is there. Okay, keep it, madam. Okay, oh, that's a new pen. Thank you. These are actin filaments. Alright, actin filaments held together by Z lines. Now they are in place. They are in place. You have to pull these two towards each other. You have to pull these two towards each other. Muzzle is like this. See, muzzles are attached to bones on either side. Skeletal muscles are attached to bones on either side. There is attachment of the biceps to the here, shoulder here, and to the elbow here. When the muzzle contracts, what is happening? Let us say the force is being generated from here. This end, the force is being generated. The first sarcomere gets shortened. So the filaments will pull the next Z line towards the previous one. What will happen to the next one? That also gets pulled. The next one also gets pulled. Next one also gets pulled. Next one also gets pulled. And all the pull here, shortening of the fibers is being transmitted to the joint. The muzzle cannot become any shorter. The muzzle cannot become shorter. The pull will be transmitted to the joint and the joint is pulled. Shortening of the muscles, because of the arrangement and design of the muscles, fibers, it will pull the joint, bringing about movement at the joint. When the muscle elongates, when the muscle relaxes and the sarcomeres return back to their positions, the pull is withdrawn and the joint goes back to its place. Joint goes back to its place. You do one thing. Hold your biceps. Feel your biceps. To feel the movement, not to feel the mass. Alright? Oh, okay. It was bigger yesterday. Feel your biceps and you flex your elbow. When you flex your elbow, biceps is becoming taut. Biceps is becoming hard. Biceps has become shortened. It is bulged. Alright. Take it back. Biceps elongates. Do the same thing holding the triceps. You hold the triceps and flex the elbow. What is happening to the triceps? It is elongating. When you extend the elbow joint, the triceps is contracting. So the joint, the elbow goes back to its position because of two things. One, extension of the biceps and flexion of the, or contraction, relaxation of the biceps and contraction of the triceps. There are always forces in play. When the biceps is contracting, the triceps is also attached to this, no? Triceps is getting pulled, triceps is extending. Alright. So it's not like the pull of the biceps is released, so the elbow falls back. It is not falling back. It is slowly, in a regulated manner, being pulled back. Alright. It is like stopping in a downhill. Acceleration is removed, but for the car to stop, somebody should apply brake, no, in a downhill. So that is what is done by the extensors. Okay. So sarcomere is the functional unit of the muscle. Sarcomere is the region of the myofibril. Sarcomere is the region of the myofibril between two adjacent Z lines. Structurally, you call it that way. It is the region between two adjacent Z lines. This is one Z line. This is another Z line. Forget about titin here. Another protein. Let it be for now. Thin lines are attached to the Z lines. Thin filaments are attached to the Z lines. And thick filaments made up of what? Thick filament is made up of myosin predominantly. Thick filament is made up of myosin, is in between the actin filaments, like this. The head of the thick filament will get attached to the thin filament and flexes during muscle contraction, pulling the thin filament towards each other. So it's like this. Thin filaments are like this. Thick filaments are attached to thin filaments in such a way that when the head of the thick filament flexes, the thin filaments come closer to each other. When the thin filaments come closer to each other, what will they pull towards each other? They will pull the Z lines towards each other, shortening the length of sarcomere. You understand? Physiology, we will look at it later, I am telling you. The function, the muscle contraction Mechanism later. Alright. So this one and this one, the thick filament and thin filament can engage with each other, disengage with each other. When it engages, the head of the thick filament will pull the thin filament. So if this is the direction of pull, if muzzle is to be pulled this way, the engagement will pull the thin filament towards this side. Alright. Thick filament is in between the thin filaments. How will it stay in position? How will it stay in position? Another connective tissue band will support the position of the thick filament, myosin filament. What is this? What you see in the middle? In the middle of the thick filament is M line. The bands are also represented by how it appears. See this. You see, here if you see, this is the, what is this? This is the Z line. This is the ultra microscopic image of the muzzle fiber. Alright. I'm 64,000 magnification. This is the Z line. This is the Z line. So this is our sarcomere. What is this zone? This entire thing? You see this? It is dark here, not so dark here. Why? Thin filaments are extending up to here, thin filaments are extending up to here, thick filament is from here to here. There is a zone in between, where there is no overlap of thick and thin filament. That zone of non-overlap is referred to as the H band. The zone of non-overlap of the thick and thin filament is referred to as the H band. The zone of non-overlap, but the zone having only thin filaments. Zone corresponding to the thin filaments, but not overlapping zone. Thick and thin filaments are not overlapping. That is the isotropic band or I band. Alright. So Z line is there, thin filaments are here and here, thick filament is in the center of the sarcomere. So thick filament is not going till the Z line. So there is a zone where the thick filament ends and only thin filament is present until you reach the sarcomere or the Z line. So this band is the what band? I band. Why is it called I band? It is the isotropic band. Appears similar, uniform. There is no contrasting appearance. A band is an isotropic band. It does not appear uniform. Where there is overlap between the thick and thin filament, it appears darker on either side of the A band. In the middle of the A band, you have thick and thin filament not overlapping, but only the thick filament being present. That is the H zone. Now mental retardation and low IQ is associated with underdeveloped brain. What all contributes to development of the brain is the next question that you have to ask. If the baby's brain has not developed, baby's body also has not developed properly, baby's brain also has not developed properly. So these are the two main problems that we are seeing in this case. What is necessary for the body's development and brain development is the next question. Absence of which will lead to declining of body's growth and brain development. What is necessary? There are two hormones. If you look at it, if you go and study back, if you go back and keep digging it, there are two hormones which will contribute to growth. What are the two hormones which contributes to growth of the body? Two hormones which contributes to growth of the body. See, all the hormones in the body are contributing to four major purposes in the body. 1. Growth. 2. Development. 3. Metabolism. for reproduction. So hormones are contributing to one of these 4 components. Some hormones are contributing to growth, some hormones are contributing to development, some are contributing towards metabolism, some are contributing in reproduction. We have growth and development issue here. Growth and development issue. So we have to look at hormones which are contributing to growth and development. Which hormones contribute to growth and development? Growth stimulating hormone. Growth hormone. Okay, growth hormone. Somatotropin. Growth hormone brings about growth of the body. Does growth hormone contribute to development of the body? Is growth hormone associated with brain development? Corollary of that is, if there is absence or decrease in the availability of growth hormone, will it lead to decreased brain development? No. So growth hormone is associated with growth. Stunted growth can be explained with growth hormone. The growth has not happened properly, maybe because of growth hormone deficiency. But we have others also. We have mental retardation and we have low IQ. Abnormal skin. Abnormal skin also can be because of growth hormone deficiency. Alright, skin not being optimally developed. Development along with growth is contributed by another very important hormone. Which hormone? Which hormone is important for brain development? Absence of this hormone will lead to mental retardation and also physical growth stunting. So growth hormone contributes to growth of the body, but this hormone that I am talking about will lead to differentiation of the body. Differentiation of the body and the body becoming more functional. So it is contributing to growth also, partially, but predominantly contributing to the development. What is ADH? ACTH, adenocorticotropic hormone. ADHD is attention deficit hyperactivity disorder. ACTH, adenocorticotropic hormone, stimulates the adrenal gland. What does adrenal gland produce? Adrenal gland will produce cortisol and adrenaline, epinephrine, norepinephrine, cortisol. Those are stress hormones. Thyroxine is correct. Thyroxine is responsible for development. Absence of thyroxine, absence of thyroid hormone during the development of the baby can lead to mental retardation. Thyroid hormone is very, very important. Now, we bring it down to the point where it can be because of non-availability of thyroxine, absence of thyroxine. Absence of thyroxine would have led to all this. Stunted growth possible, thyroxine not available, stunted growth is possible. Mental retardation and low IQ definitely because of thyroxine absence. Nothing else contributes as significantly to thyroxine in mental development of the baby. What can lead to low thyroxine is the next question. Okay, this is possible because of low thyroxine, thyroid hormone deficiency in the baby. What can lead to thyroid hormone deficiency in the baby? Most common possibility is low iodine in the diet of the mother. Baby has to be supplied with iodine so that the baby's thyroid gland will start producing thyroxine in adequate quantities and the baby will have all the benefits of what thyroxine gives. Baby cannot get anything other than what the mother gives the baby. So if the mother's diet was deficient in iodine, there is a possibility that the baby did not get enough iodine for its development. There is a mechanism in the body. When it comes to shortage of resources in the mother's blood, the baby's interest will be prioritized by the body over the mother's interest. So if iodine deficiency is there in the diet of the mother, whatever available iodine is there will be pushed for the baby's utilization. Placenta will pull it off and push as much as possible to the baby. So who will experience iodine deficiency more? Iodine deficiency impact will be more on whom? The baby or the mother? Mother. It will be more on the mother. So if the baby is having iodine deficiency, it will be seen in the mother also. Thyroid testing is therefore very important during pregnancy. If the mother is showing deficiency of thyroid hormones, then the baby also might be experiencing that. The baby will be experiencing lesser deficiency than the mother. So iodine supplementation to the mother will take care of the baby's needs also. So that is possible. Deficiency of iodine in the diet is possible. But we have another option, cancer of the thyroid gland. Cancer of the thyroid gland is another option. We know the problem in the baby is because of the thyroid gland only. Thyroid gland malfunctioning. It can either be because iodine is not enough, so the baby has not got enough iodine, thyroxine is not produced in adequate quantities. Can it not also be because the thyroid gland of the baby is cancerous? Maybe, possible. But well, between a possible less likely situation or option to a more commonly found option, which one do you want to choose? The common possibilities, the common cause is what you have to choose. I cannot say no for option 3, cancer of the thyroid gland. Cancer of the thyroid gland of who? That is not mentioned, no? It is not mentioned whether it is the cancer of the thyroid gland of the mother or the baby. So there also we don't have enough information to conclude. If it was mentioned as the cancer of the thyroid gland in the baby, then possible. If it is a cancer which is not producing hormones, making the thyroid gland malfunctional, then this will happen. Cancer can also make the affected organ produce a hyper production of whatever it is supposed to produce. Thyroxine can be produced in excessive quantity. May not be very proper in its structure and function, but it can be produced in excessive quantity also if it was cancer. So the reverse is also possible. Hyperthyroidism symptoms are also possible with cancer of the thyroid gland. So cancer of the thyroid gland is less likely to be the right choice here because it is not mentioned whether it is of the baby or the mother. Even if it was mentioned that it was of the baby, even then we cannot be sure that that is what will lead to what is leading to hypothyroid situation. It can lead to hyperthyroid situation also. If option A was not there, instead of deficiency of iodine in diet, if the option was, let's say, excessive production of insulin, then we can go with option 3. Then we can go with option 3 because that is the one which is relating to thyroid. Possible that during cancer of the thyroid gland, there can be hypothyroid situation. Even though information is not given, we could have assumed that it is relating to the baby because there is nothing else which will correlate. Iodine deficiency is the clear choice here. Alright. Deficiency of iodine in the diet. The symptoms described are indicative of a condition called congenital hypothyroidism, or congenital hypothyroidism is referred to as cretinism. Cretin is also a dwarf. So you have two types of dwarfs that you can see because of hormonal insufficiency. A hypothyroid dwarf and a pituitary dwarf. Hypothyroid dwarf and a pituitary dwarf. Pituitary dwarf is because of low production of growth hormone. When growth hormone production is low, you will find pituitary dwarfs. Alright. So, have any of you seen Game of Thrones? You find that dwarf. What's his name? I forgot. Huh? Tyrion Lannister. Tyrion Lannister is cretin or pituitary dwarf. He's a pituitary dwarf. He doesn't seem to have mental retardation. He seems to have hyper intelligence. Okay, right. Okay. Iodine deficiency. This explanation I have given, and because see, iodine is not normally and commonly found in the plant-based foods. Iodine has to be, that is why, and iodine deficiency, hypothyroidism was very prevalent in our country few decades ago, leading to iodine deficiency goiter. Iodine deficiency goiter. Thyroid is a very important hormone for maintaining metabolism. Your basic metabolic rate is maintained by thyroid hormone, thyroxine. And basic metabolic rate is what will keep your metabolism in good pace. If sudden weight loss, hypothyroidism can lead to weight gain. Why? Hypothyroidism will reduce the metabolic rate. Metabolism is what? Burning. It's like the internal combustion engine of your body. Alright. It will use the fuel and burn, produce heat and energy for your bodily functions. If that fuel burning is reduced, fuel is stored, accumulation of fuel is what is weight gain. Burning of more than necessary fuel is what is weight loss. Reserves are being preserved, weight gain happens. Reserves are being excessively removed, weight loss happens. So thyroid is very critically connected with our normal living. So because iodine deficiency was the most common cause of thyroid malfunction, it was decided that salt will be fortified with iodine. Fortification. So iodine is added to table salt. Earlier when we were in our younger days, we used to get iodized salt and non-iodized salt also. So it would be clearly mentioned on the packets, iodized salt. Now all salt is iodized salt, whether it is powdered salt or crystal salt. It is compulsory to fortify all salt which is used for consumption. Whatever humans are supposed to consume, iodine fortification is done by default to all salt, whether it is powder or crystal salt. So that took care of a populational problem. Large number of people could overcome the problem of iodine deficiency goiter. Right. There is one more goiter, iodine deficiency goiter is hypothyroid goiter. There is one more disease where hyperthyroidism causes goiter, problem, where you will find exophthalmia. There also you find goiter, hyperthyroidism, hyperactive thyroid gland. Disease is called Graves' disease. Graves' disease, no? Not studied? Graves' disease is a hyperthyroid abnormality disease. We will discuss that when a question comes particularly about thyroid gland. It causes burning of fat, rapid burning of fat. The intraocular pad fat is burnt, making the eye sockets go inwards. The eye socket is properly positioned. The tissue around the eyelids and all the soft tissue around is properly positioned to make our eyes look appropriate in terms of the size. If you burn the fat around it, all this skin and the lids and all will get retracted. They will get retracted when the cushioning behind them is removed, it gets retracted and the eyeballs appear like they are popping out. Exophthalmia. Somebody will have like big eyeballs, large part of their sclera is visible. Exophthalmia. You will find very restless and anxious personality. Such people will be anxious personality or very restless, can't they keep fiddling with things, can't stay calm. Hyperthyroidism causes all that. See, your behavior, your responses, your action, reaction, everything is dependent on the chemical play. Have you seen this movie, Lageraho Munnabai? No? In Lageraho Munnabai, he tells, no, Munna Bhai tells, chemical locha. He is able to see Gandhiji because of chemical imbalance in the brain, hallucination and all that. So, our life is also chemical locha only. You maintain chemicals properly, things will be good. Influence of alcohol, any sane person will become insane with alcohol. Why? There are checks and balances. Chemicals again. So we assume that we have a lot of control on how we think, how we behave, what we do and all that. But chemicals are controlling. All that we have to do is ensure that the chemical balance is appropriate in our body. You take care of the chemical balance, body knows what to do. Alright. So, I'll give an overview of the hormonal ecosystem in the body. Hormonal ecosystem in the body, yes. Yes. Go. No, that's the standard operating protocol now for me. Anybody wants to use the washroom, I'll just make them get noticed and then send them. It's okay. Endocrine glands and corresponding hormones. Alright. Gland, where is it present? What are the hormones produced? What does the hormone do? Some basic things only, not in great detail. Pituitary gland is present at the base of the brain. Anterior pituitary, growth hormone, stimulates growth of bones and tissues, regulates metabolism. Alright. Thyroid stimulating hormone. See, anterior pituitary produces a lot of tropic hormones. What are tropic hormones? Hormones which will stimulate something else to do their work. Alright. They are not like the people who work, they are like the managers. Manager's job is what? Make people work. Manager will only tell, hey, you do this, you do this, you do this. What are you doing? I am telling you to do it. My job is to make you do. Alright. So, growth hormone, thyroid stimulating hormone, adrenocorticotropic hormone, follicle stimulating hormone, luteinizing hormone, prolactin. These are the anterior pituitary hormones. If you see, 3 hormones, FSH, LH and prolactin, are associated with reproductive functions, stimulating the reproductive function. ACTH is associated with stimulating the adrenal gland, which will control the sympathetic responses. TSH will stimulate the thyroid gland, which will regulate metabolism. Growth hormone will stimulate receptors in various parts of the body, which will bring about growth and development. In all parts of the body, that brings about growth and development. Posterior pituitary, if you look at it, ADH, antidiuretic hormone, regulates renal function. Oxytocin regulates smooth muscle contraction in various parts of the body. Predominantly seen in its effect on uterus and the childbirth during pregnancy. There is one more, there is one middle lobe of the pituitary gland. What does it produce? Which is not produced in human beings, that is why I have not given it here. There is one hormone produced by the middle lobe of the pituitary gland. MSH. MSH is melanocyte stimulating hormone. What are melanocytes? Melanocytes are the cells which are responsible for skin pigmentation. We also have melanin containing cells. Are they not melanocytes? Are the cells that we have in our skin not melanocytes? Why don't we need melanocyte stimulating hormone? Our melanin pigmentation is dependent on the genetic combination that we have. It is a permanent structure that is developed. Melanocyte stimulating hormone is an adaptation feature. Melanocytes are adaptive cells in organisms which have it, like frogs or other organisms which have melanocytes. It will help them in camouflaging and it will help them in better survival by altering the pigmentation in their cells. Alright, we don't need that. we have lost it over time because we have developed mechanisms of staying protected at all times. We don't need to camouflage, the other animals need to camouflage and escape from us. We are at the top of the pyramid. We are at the top of the pyramid, but we are not a part of any food chain. If you understand it, we are at the top of the pyramid of life. But because we don't have a pattern of food, we are not in any food chain. Alright? We can get into any part of the food chain. People, if they have access, they wouldn't mind eating lions and tigers also. Okay, taste. Alright. So thyroid gland is below the neck. It's called below the Adam's apple. Adam's apple is what? What cartilage is that? What cartilage? Sound box, larynx. Larynx. It is Adam's apple is the larynx, sound box. T3, T4 is produced. Which is the active hormone here? T3 or T4? Both will have its functions. But which one is the predominantly functional hormone of the two? T3. T3 is more active. T3 is more active. So it regulates metabolism, growth and development. Then you have calcitonin. Calcitonin and parathyroid hormone, thyroid, parathyroid. Along adjacent to the thyroid glands, on either side you have parathyroid glands. Parathyroid gland produces one hormone called parathormone or parathyroid hormone. Calcitonin is produced by thyroid gland. Both of these, the job of both these hormones is to ensure calcium level in blood. Why should there be appropriate levels of calcium in blood? Muscle activity is dependent on calcium. So providing adequate concentration of calcium in the blood is necessary for skeletal muscle function. So you have to maintain a level of calcium in the blood. Where will calcium come from? You have to absorb it from the diet. If calcium has to come into the blood, you have to absorb it from the diet. You have to reduce elimination of calcium from kidneys. 3 places where you can control calcium levels. One is intestine. You can regulate absorption. Second one is elimination at the level of kidneys. You can manipulate that. If you increase elimination, calcium levels will come down. If you decrease elimination or you block elimination of calcium, calcium levels can be raised. Third one is taking from the bank. Which is the calcium bank in the body? Bones. So if you want to increase calcium, you will have to break the bone. Correct? If you have excess calcium. Now let us say, if you want to increase calcium in the blood, what will you do at these 3 levels? Increase absorption, decrease excretion, increase resorption of the bone. If you want to decrease calcium level in the blood, what will you do? Decrease absorption, increase excretion or increase accumulation, increase storage. You push it to the bone. If you want to decrease calcium levels in the blood, you can push calcium into bones. If you want to increase calcium levels in the blood, you will pull calcium from the bones. Calcitonin and parathormone are opposite. Calcitonin is like the bone making hormone, parathormone is the bone breaking hormone. So what will calcitonin do to calcium levels in blood? Calcitonin, what will it do to calcium levels in blood? Lowers calcium level because it is bone making. For that, what will it do to blood calcium? It will push it into bones. Parathormone is bone breaking hormone. What will it do to calcium levels in blood? It will increase. So parathyroid gland is that. Then adrenal gland, it is on top of each kidney. There was one lame joke that used to go around in our medical college when we got introduced to all these concepts. Kidneys are Gandhians. Kidneys are Gandhians. Why? We have two kidneys. Kidneys are the Gandhian organs of our body. Why? Because kidneys are wearing a cap. I told you, lame joke. What is the cap? Adrenal gland. Doesn't it appear like that, like Gandhi cap? So, you can't take off those kind of lame images. If those lame explanations transform into images in your brain, you can't remove that. It will stay. You will always recall it. You will not tell people how you have registered it. You will let people feel that you have great memory. Alright? So it's a memory tool. You have to remember it you can remember it in the most disgusting way. As long as it works for you, there is no restriction on how you correlate it and remember it. However you want to remember. But it should help you recall. Make sure that you don't tell people what is the connection that you have built for memory. That is for your own, to maintain your own image and reputation. That's all. Who cares how you build concepts in your mind, as long as you can understand it and use it when you want it. Problem comes because image management has become too personal. Image management or maintaining that particular persona is for public consumption. If you make it too personal, even in your mind, you will start having that that layer of pretense. You should have your own space psychologically where there is no pretense. You are not pretending. You are just being as you want to be. In a social environment, in a social situation, you can't do that. People tell that they are the same whether somebody is there or not there. That's not possible. It's not possible. Because in a social situation, you have to act appropriate to the situation. When it is in a private situation, your own personal space, there is no appropriateness. What is comfortable for you is the appropriateness. So, while you are studying, your mind is your private space. How you build concepts, how you understand, how you correlate, all of that is your choice. Whatever connects to you. But when you are expressing it, that is the public situation where you have to think and express it in a way that it's appropriate to the place where you are and to the people who you are talking to. That's all. So don't make pretense or image management too personal. It will take away your happiness. Adrenal cortex, cortisol, aldosterone, androgens. Adrenal medulla produces epinephrine, norepinephrine. Pancreas is a dual organ. It's a mixed organ. It has both endocrine and exocrine components. The endocrine part is the islets of Langerhans. You have insulin, glucagon, major hormones produced, and there is one more hormone produced which will regulate the digestive hormone, digestive enzyme production, stimulation. It regulates, which is that hormone. The third one which is produced from islets of Langerhans of pancreas. Insulin is produced from beta cells. Glucagon is produced from alpha cells. This one more is produced from delta cells. Somatostatin. No? Not studied? Somatostatin. Alright, somatostatin will regulate gastrointestinal hormones. We will see that when we come there. This we will continue tomorrow again. Pineal gland is very important. It is present in the center of the brain. It is also referred to as the third eye. Just behind this, if you go straight, you will reach pineal gland. Pineal gland produces melatonin. Melatonin is responsible for what? Melatonin is responsible for sleep-wake cycle. Okay, I'll ask you something. Increase in the level of melatonin production causes sleep or wakefulness? Sleep? Are you sure? Sure? Okay. When you wake up in the morning, the levels of melatonin will be low. The temperature inside the brain will be low. Alright. As the day progresses, because brain is a hyper metabolic organ, it produces a lot of heat. Metabolism is very aggressive. It consumes a lot of energy for its metabolism. So heat production will lead to increased metabolism, increased carbon dioxide production, fatigue in the brain cells. And as the day progresses, related to the temperature, as the temperature on the outside keeps increasing, as the light keeps increasing, not temperature, light, melatonin production keeps on increasing. So by the end of the day, melatonin would have reached a threshold which will stimulate sleepiness. Where is that activated? Sleep and wakefulness is associated to which part of the brain? RAS. What is it? Reticular activating system. Oh, we'll come there. Reticular activating system is associated with sleep-wake cycle. So it will suppress the reticular activating system and inducing sleep. So if you want to manage sleep properly, two things you have to, whenever you sleep, remember, whenever you sleep, melatonin will be cleared. Sleeping will initiate the mechanism of clearing melatonin from the brain. Alright, and temperature management, head. Pranayama, Nadi Shodhana Pranayama you do, no? The Nadi Shodhana Pranayama that you do and the Bastrika Pranayama that you do, both of that will help you in cooling down the brain. It cools down the brain and it removes carbon dioxide accumulation. It hyperoxygenates, increases the clearance of carbon dioxide, removing the metabolites which will stimulate fatigue in the brain, increasing your wakefulness. Alright, and sleeping will cause melatonin to get wiped off, get removed from the brain cells. That is why sleeping during the day, learning to sleep 15-20 minutes, not more than that, 15 to 20 minutes, practicing to sleep for 15 to 20 minutes during the daytime is a great power for improving intellectual productivity. Alright, it is not how long you can work, it is how well you can rest that will determine how productive you will be in the way you work. Everything of your routine, every small bit is designed. It is not just written from somewhere. It is designed. So practice sleeping for 15 to 20 minutes. You will know when you are going for exams. Let's say you are 1:45, you are supposed to be in the examination hall. You are allowed to go there in the examination hall. You will start writing at 2 o'clock. Correct, no? You will start writing at 2 o'clock. If you have practiced resting for short duration, as you are sitting in the examination hall, you can just close your eyes, do pranayama, the 2 pranayamas that you are practicing now. Bastrika you cannot do, you can do alternate nostril breathing, you can do deep breathing. You have to gain certain extent of mastery over that breathing pattern. Then it will start giving you benefits. That 5 minutes of regulating the breathing will bring about so much focus in your mind. It will take away the fatigue, it will bring focus, it will reduce anxiety. You can hit the ground running. To start the exam with anxiety, with tension, and settle down eventually and perform well. Even if you lose 10-15 minutes in the examination hall to settle down from your anxiety, it is a great loss. Those 10-15 minutes you are not in your full focus. The thought of having lost 15 minutes to settle down in your anxiety would make you hurry, would make you rush. And in that rush, you are likely to make mistakes, judgment errors happen. So small things. final performance is dependent on a lot of things that can happen spontaneously without you actually consciously doing it. And something will happen spontaneously if it becomes a part of your habit pattern. If it becomes a part of your habit pattern and your behavioral structure. And that can happen only with consistent, continuous practice for a sufficient period of time. We have enough time, 2:55. Alright, let's practice. Endocrine system, we will continue discussing in the next class tomorrow. We will, because endocrine is the regulating system for all the systems of the body, we will be touching upon multiple systems through endocrines, from the perspective of endocrines. Alright, so tomorrow we'll do this.